The Opisthokonta Greek, Opisthios.
Rear, posterior.
Plus kontos. Pole, i.e. flagellum, are a broad group of eukaryotes, including both the animal and fungus kingdoms. The Opisthokonta, previously called the fungi metazoa group, are generally recognized as a clade. Opisthokonta together with Apusimonidida and Breviata comprise the larger clade Obizoa.
Flagella and other characteristics.
A common characteristic of Opisthokonta is that flagellate cells, such as the sperm of most animals and the spores of the chytrid fungi, propel themselves with a single posterior flagellum. It is this feature that gives the group its name. In contrast, flagellate cells in other eukaryote groups propel themselves with one or more anterior flagella. However, in some Opisthokonta groups, including most of the fungi, flagellate cells have been lost. Opisthokonta's characteristics are including synthesis of extracellular chitin in exoskeleton, cyst, spore wall, or cell wall of filamentous growth and hyphae, the extracellular digestion of substrates with osmotrophic absorption of nutrients, and other cell biosynthetic and metabolic pathways. Genera at the base of each clade are amoeboid and phagotrophic.
History.
The close relationship between animals and fungi was suggested by Thomas Cavalier Smith in 1987, who used the informal name Opisthokonta. The formal name has been used for the chytrids by Copeland in 1956, and was supported by later genetic studies. Early phylogenies placed fungi near the plants and other groups that have mitochondria with flat cristae, but this character varies. More recently, it has been said that holozoa animals and holomycota fungi are much more closely related to each other than either is to plants, because Opisthokonta have a triple fusion of carbamoyl phosphate synthetase, dihydrooroutase, and aspartate carbamoyl transferase that is not present in plants, and plants have a fusion of thymidylate synthase and dihydrofolate reductase not present in the Opisthokonta. Animals and fungi are also more closely related to amoebas than they are to plants, and plants are more closely related to the SAR supergroup of protists than they are to animals or fungi. Animals and fungi are both heterotrophs, unlike plants, and while fungi are sessile like plants, there are also sessile animals. Cavalier Smith and Stechman argue that the uniciliate eukaryotes such as Opisthokonta and Amoebozoa, collectively called uniconts, split off from the other biciliate eukaryotes, called biconts, shortly after they evolved.
Taxonomy.
Opisthokonta are divided into holomycota or nucleomycia fungi and all organisms more closely related to fungi than to animals, and holozoa animals and all organisms more closely related to animals than to fungi. No Opisthokonta basal to the holomycota-holozoa split have yet been identified. The Opisthokonta was largely resolved by Torriella et al. Holomycota and holozoa are composed of the following groups. Holomycota fungus-like. Fungi includes Nuclearida, chytrids, flagellated, zoosporic fungi, Microsporidia, previously thought to be sporozoans, Hyaloraphidium, previously thought to be a green alga, now considered a fungus, Fonticula excludes Oomocetes, water molds, now included in the SAR supergroup. Labyrinthulomycetes, slime nets, now included in the SAR supergroup. Myxomycetes, now included in amoebozoans. Rosellida, placement uncertain. Holozoa, animal-like. Mesomycetozoa. Dermocystida, formerly considered parasitic fungi or sporozoans. Ichthyophonida, formerly considered parasitic fungi. Incerticides. Ecrinales, formerly considered trichomycetes. Amoebodiales, formerly considered trichomycetes. Corallichytrium, formerly considered a heterocont. Phylozoa. Pilastria. Choanoflagellata, flagellates formerly included in protozoa. Animalia, including Myxozoa, previously considered fungi, now considered cnidarians.
Phylogeny.
The choanoflagellates have a circular mitochondrial DNA genome with long intergenic regions. This is 4 times as large as animal mitochondrial genomes and contains twice as many protein-coding genes. Corallichytrium seem likely to be more closely related to the fungi than to the animals on the basis of the presence of ergosterol in their membranes and being capable of synthesis of lysine via the AAA pathway. The ichthyosporians have a 2-amino acid deletion in their EEF1A1 gene that is considered characteristic of fungi. The ichthyosporian genome is greater than 200 kilobase pairs in length and consists of several hundred linear chromosomes that share elaborate terminal specific sequence patterns. In the following phylogenetic tree, it is indicated how many millions of years ago the clades diverged into newer clades. The Holomycota tree is following Tedersu et al. One view of the Great Kingdoms and their stem groups.
Gallery. References. External links.
Tree of Life Eukaryotes.